Hello, everyone. Thanks for coming to Bio at Noon again. It's been a while since we've all gathered, and I'm very pleased to, oh, good job, Victoria. She put the recording on already, to introduce Sophie Clayton this week as our Bio at Noon speaker. Sophie did her bachelor's degree in Bangor University in the UK, and then did her PhD at the MIT Hui Joint Program. Sophie is now an assistant professor of oceanography at Old Dominion University. And if you have just joined us, questions today will be fielded. You can put them in the chat if you'd like, and Sophie will answer them after she stops giving her seminar. Or if you'd like to unmute yourself and ask questions during the seminar, please feel free to do that. Sophie said that's a great way to have a discussion during seminar. So without further ado, I will hand over the screen to Sophie.
Okay, well, hopefully I don't mess up screen sharing. I feel like that's the first test, right? I need you to enable me. The host, Heather, you need to give me the power. Okay, let's try again. So,
cool.
All right, so I think you should now be able to see my slides, and amazingly, I can actually still see you. Wonderful. Wow. Right. I'm going to look like I'm looking off to the side. I'm working with two screens. So the screen with my slides is over to this side. So it's not that I am ignoring everyone. It's just that I'm working in this two-screen world that's becoming more and more normal. Thank you for inviting me. And Heather's invitation was so lovely. I couldn't refuse. And I'm very glad to virtually talk to you. I'm glad we've managed to sort of find ways to keep having seminars and exchanging ideas during this lockdown period. So thank you guys. So I want to talk about some observations that I made with several colleagues listed on this title slide, actually 6 years ago now in the California current. And we made measurements of various things, including trace metals, dissolved nutrients, photophysiology, chlorophyll, of course, as well as DNA to look at phytoplankton community structure. And it was somewhat of an ad hoc study. It was not intended, but we discovered a couple of mesoscale eddies near our study site and had some time to survey those. So I want to talk to you guys about that work. And I do want to acknowledge particularly Drew Chappell. So a big chunk of the work that I'm going to present today actually came out of her lab and was completed by her student, Susie Abdallah. And I've been working very closely with Fedor Kuzminov, Claire Till, Tyler Cole on a lot of the nutrient and trace metal data that I'm going to show you guys today. So this is work completed by many people other than myself. So hopefully, if I say anything stupid, you can attribute that only to me and not to any of these other people who have done some great work on this project. Okay, so I'm going to talk about the California current system, and this is just to give you guys an overview. Here is sea surface temperature. Hopefully you guys are familiar with the California current system. It's an eastern boundary current that has upwelling along the coast, and the upwelling is pretty seasonal. So in the summertime, you tend to see stronger upwelling that's bringing up cold, nutrient-rich waters along the coast, which drives quite high productivity. So the upwelling tends to bring up dissolved iron as well as nitrate and other dissolved nutrients. And then it moves offshore out into what's called the transition zone here before then sort of we get out into an oligotrophic California current region further offshore. And the idea is that you have these upwelled nutrients and trace metals quite near the coast. You have a big pulse of productivity, but then that sort of tends to tail off as you move offshore. And this transition zone is dominated by mesoscale eddies, and they're the mechanism by which you have this communication between the coastal zone and the offshore oligotrophic California current region. And within the transition zone, you have several processes within the eddies whereby nutrients are being drawn down, community structure may be shifting before you get out into that completely different sort of oligotrophic regime that's over out here. So there's been various work done by people in Ken Brulin's lab and others looking at iron upwelled near the coast, along with other micronutrients and macronutrients, and how it sort of changes, gets depleted through this transition zone. So this study, the cruise at least, was intended to continue with that work, looking at processes of upwelling of trace metals and community responses. But I was quite lucky in that we had a bit of a pause in the upwelling. So most of the people on the cruise were quite sad because they were there to study community responses to upwelling along different parts of this coast. But I'm not going to say I was happy because that would sound mean, but it gave me a wonderful opportunity to go and survey some eddies. So we found 3 mesoscale eddies close to the study site. And because we had this pause in the upwelling, Ken Bruland, who is the chief scientist on this cruise, was happy to kind of hand over the ship to me for 3 days so that we could go and do some studies across these features. So here's a map of sea surface height, and we'll come back to this, so don't worry too much about the details right now, but these gray contours are showing the sea surface height anomaly. I'm showing chlorophyll as the background color, and then in black, the cruise track. And you can see we spent a lot of time hanging out by the coast looking for upwelling. But as I said, we had this opportunity to come out into the transition zone to survey 2 cyclonic eddies. So this is one that's further offshore and one that's further inshore that I can show you guys in a minute, and an anticyclone that was further to the south down here. So we took a few days off some of the culture work and closer to the coast, sort of, not culture, what I'm looking for, incubation studies that we were doing near the coast, and came out to do some surveying. And this was in July of 2014. It was kind of an interesting year because the blob was sitting offshore in the Pacific, kind of shutting down some of the more energetic upwelling that you might have expected for that time of year. So we did this opportunistic but extensive survey of these mesoscale eddies. Okay, so a bit more detail about these features. Again, the gray contours are sea surface height anomaly. So you can see here right by the coast, we have a cyclone and another larger cyclone that's offshore. So I'm going to stick with this naming throughout the talk. And I've actually got these the wrong way around on this slide. That's embarrassing. This should be C1. So C1 is a younger cyclonic eddy that had been formed roughly a month prior to us sampling it. And then C2, this larger, older cyclonic eddy, which had formed kind of in the same location as C1, but 10 months prior to sampling. So we have these 2 cyclonic eddies formed in roughly the same place, but several months apart. We also sampled an upwelling plume here. So we sampled along this sort of plume of colder waters that was coming off the coast along here, and that was only from the surface. So this is underway sampling. And then finally, we came down here and sampled this anticyclonic eddy that was also pretty new. It had been formed around 1 month prior to sampling. So we have these 4 features. So in the upwelling plume, we estimated the upwelling probably started about 2 days before we sampled it. Then in C1, ignore this, in C1, roughly 1 month old. This older eddy, 10 months old, and then the anticyclone was roughly a month old. So we have these 4 features that I'm going to talk to you about in more detail. Okay, so the idea behind the study and the things that I'm hopefully going to convince you of or talk to you about today. First of all, we want to look at the biogeochemical properties of these features to see if we can translate that into a kind of time evolution of these biogeochemical properties from freshly upwelled all the way out through the transition zone to the oligotrophic sort of California current end member. We're also interested to see if we saw any bulk community response. And when I say community response, I'm talking about the phytoplankton. So I apologize, I'm a bit phytoplankton centric. So I'm talking about the phytoplankton community response here to the way these biogeochemical properties are evolving across that upwelling transition zone California current continuum. Then I'm going to talk about some work where we zoomed in onto the phytoplankton community, and we wanted to see how they were varying across these features. And this is really only looking at the cyclonic eddies. So we're comparing C1 and C2, the young and the old cyclonic eddies. And then finally, there's some sort of tantalizing evidence that we might be seeing some submesoscale, fine scale effects in the biogeochemistry at the edge of these cyclonic eddies, but that's a little bit more speculative. Okay, so I didn't really tell you much about the measurements that we made, but here's a rundown. So C1, the younger eddy, and C2, and the anticyclone, and here we have this upwelling filament. So we did a series of eddy surveys. So these open black circles show places where we had CTD stations. So we have roughly 5 to 6 for each of the cyclonic eddies. We have 5 across the anticyclonic eddy. And they're about 20 kilometers apart. So this is a pretty synoptic mesoscale survey. We managed to cross through each of the eddies in less than 24 hours. I think it was closer to 18 hours in each case. And we took measurements for dissolved nutrients, trace metals we got in only a subset of the CTD stations, so at the edge and at the center of the eddies. We did collect samples for DNA and flow cytometry, but I'm not going to talk about those today from the CTD casts. And we also had chlorophyll samples. So that's the depth resolved sampling that we did. Then we also did a fair amount of continuous underway sampling. And so these red lines show you the tracks where we completed this continuous underway. So that's surface only sampling across these features. So the upwelling plume, we really only did a surface survey. The cyclonic eddies here, we did a combination of CTD surveys and underway sampling at the surface. And then for the anticyclonic eddy, we did just a CTD survey across that feature. So for the underway sampling at the surface, we also got samples for dissolved nutrients and trace metals. Fedor was running a fire system, so we have photophysiology. We got FV over FM estimates from those surface underway sampling. And then Susie collected, actually Drew collected DNA samples that Susie then worked on. And we also have chlorophyll from these, from the surface sampling. Again, we have flow cytometry, but I'm not going to talk about that today. So we've collected actually a pretty extensive data set across these features, which is super exciting, but also a lot to try and digest. So I'm going to show you some of the results, some of the things that we observed. I have some ideas about what it means, but hopefully you guys will too. So I'm looking forward to some feedback from you guys about how to deal with this wonderful data set that we've collected. Okay, so first of all, I'm going to talk about the biogeochemical properties across the features and try and answer this question of whether or not we're seeing some kind of phytoplankton community response. So I'm going to start up with the underway sampling. So I'm going to show you the results of what we saw underway from the 2 cyclonic eddies and this upwelling plume. So just a quick reminder, this was fresh upwelled water that we sampled along this line. It's a little bit, I probably should have shown this on SST. If it was plotted on SST, you would see really clearly a cold plume of water following that line. So this freshly upwelled water, roughly 2 days post upwelling, and then this 1-month-old cyclone and 10-month-old cyclone. Right, so there's a lot on this slide. So just for reference, I'm showing you data that was collected at the surface along this upwelling plume. So temperature here in black, you can see sort of close to 10 degrees near the coast. So this is near the coast, this is going offshore. You can see temperature gradually increases. So towards the end of this transect, we've kind of dropped out of the upwelling plume. Fluorescence here in green, we have very high fluorescence right near the coast in that freshly, or relatively freshly upwelled water a couple of days after the fact. And then it drops off pretty abruptly as you move offshore, slightly elevated for a while along here, but then as you get into the warmer waters, it's really dropped right down. So now looking at dissolved iron in blue and silicate is in red. You can see both dissolved iron and silicate are elevated near the coast, so in that freshly upwelled water with the high fluorescence. And then iron drops off very quickly, but kind of wiggles around a bit. I don't know what a technical term would be. I could say there was some submesoscale variability in the dissolved iron. Maybe that sounds better. So we do see some variability in dissolved iron, but then it drops off really quickly as we're moving offshore. Pretty much the same thing for silicate, although there's a much less abrupt drop, and that sort of continues along before really dropping down close to zero as we get offshore. So then in this last panel down here, the bottom panel, I've plotted silicate in red again, just for reference. And then in black, I'm showing you nitrate. So I think the big difference that I notice here is that the nitrate drops down much less quickly than silicate. So we're seeing differential drawdown in these nutrients. We see that the dissolved iron really gets taken up quite quickly as we move along this plume. Silicate then follows, but nitrate seems to stick around for a fair bit longer than either the dissolved iron or the nitrate. So this is just following this sort of relatively freshly upwelled water along this upwelling plume here. And we can already see that there's differential drawdown in these different nutrients. So now I'm showing you data from the surface from these two cyclonic eddies. So C1 here is this young 1-month-old cyclone. C2 is the older 10-month-old cyclone. And again, this is just data collected at the surface along this red line. And I want to just note that there are different scales compared to the previous slide, particularly for fluorescence. So on the previous slide, fluorescence was going up to 20 relative units, whereas here it's topping out at 3. So there's quite a big difference there. And then this gray line roughly marks the sort of midpoint between these two cyclonic eddies. So in C1, chlorophyll is quite variable, and we do see our peak value in this younger eddy, and it sort of drops down in between the two cyclones, which you would probably expect where the water's getting warmer. So this is probably this intermediate region between the two. And we do see that it picks back up in the second cyclonic eddy, C2, and it's very much skewed towards the left side of that feature. And in terms of temperature, we can see again, temperatures are generally cooler in the younger eddy, and they've increased up to almost 17 C in the older cyclonic eddy. So we're definitely looking at some pretty different water masses there. So again, I'm showing silicate in red, and it's repeated on both of the figures. Dissolved iron is in blue. So dissolved iron is sort of on average is higher in C1 than it is in C2. But we do see this strange uptick in between the two eddies. I'm not entirely sure what's going on there, but this could be advective. So there could be a source that's actually sort of wrapping around between these two cyclonic eddies. So this is something that I'm not sure about, but I want to look into some more. Silicate again is higher in C1 and then drops down considerably into C2. And same, nitrate is considerably higher in C1 than in C2. So we have, these two cyclonic eddies that geographically are very close together, but we know that they were formed in very different time periods, even though it was in roughly the same place. And biogeochemically, they have quite different properties at the surface. So I guess one takeaway I'd say already is, not all eddies are created equal, even if they're right next to each other. They might look quite different when you take a closer look at them. So C2 is definitely much more oligotrophic than C1. There's potentially some evidence of dissolved iron supply by these eddies. We see the sort of uptick in iron here in C1. We see maybe a bit of an uptick in C2. I'm not completely convinced. In this case, silicate and nitrate co-vary, so we don't see that sort of differential that we saw along the upwelling plume. They're much more correlated than they were along the upwelling plume. And as I said, C2 appears to be much more oligotrophic in the surface, so already quite different. Okay, and so I asked this question about community response, and we collected quite a lot of FE over FM data and also data on the cross section. I am not very well versed in photophysiology, but Fedor and I have had several conversations about this data. So I'm showing you here a kind of an amalgam of FE over FM data from the tracks that we covered across those features, but I'm only looking at data that was collected at night to get rid of any sort of effects I would get from looking at it in the daytime. So this is not data that corresponds exactly to those features, but it's sort of roughly subset into regions that correspond to them. So it's not exactly along those surface tracks that I showed you in the previous maps. That said, we do see some features. So I've color coded all of these points by temperature. So 10C down here is sort of pretty freshly upwelled cold waters. And then as we get out into the warmer California current waters, we're up to almost about 17. And you can see that over here. And I'm showing you longitude along the x-axis. So the coast is over on this side. We're going offshore out here. And then I've added some gray vertical lines, just to sort of roughly subset into the regions that we've been talking about. So we see a lot of variability in FE over FM in this sort of upwelling zone, but we do see the highest FE over FM values of 0.6 out here. And that roughly corresponds to happy diatoms. So I've been told. So this, this looks like this is a region where we have a relatively happy growing community that are not particularly stressed for iron or nutrients. There is some variation, and that's just to do with the way I pulled the data together and the fact that we spent some time going up and down the coast when there was and there wasn't upwelling happening. So some of this variability is sort of can be explained by taking a closer look at the data. But we do see these highest FE over FM values in this freshly upwelled water. And then as we move offshore, we see, generally the trend is for FE over FM to decrease, but then we see variability within these features. So in C1, it's still pretty high over at the eastern edge towards the shore and then drops down to much lower values. But then we see an uptick in between the two eddies. And then again, a further drop as we get out into C2. And then, interestingly, as we move out of C2 into the oligotrophic California current waters, we see an uptick again. So what we're seeing with the FE over FM data is sort of a combination of iron stress, nutrient stress, but also community composition. So some of these higher values nearer the coast, we can sort of say this is we're looking at happy diatoms who have plenty of iron, plenty of nitrogen. They're growing quite happily. And we see this uptick over here, even though we're in an oligotrophic regime, we're actually seeing higher values because we've had a complete community shift towards more cyanobacteria. So we've got a lot more Synechococcus and Prochlorococcus sitting out here in this warmer waters out offshore. So I'm still sort of trying to work out how to interpret some of the photophysiological data, but generally speaking, we're seeing this sort of general decrease moving away from the coast. And some of the details on top of that are probably to do with patchy nutrient inputs, as well as shifts in the community structure that are just giving us sort of different baseline FE over FM values to work with. Okay, so now I want to show you some data from the eddy surveys just to give you this vertical sense of what's going on. It doesn't really change the picture much, but there are some interesting details. So I'm going to show you sections across C1, C2, and AC, the anticyclonic eddy. So this is the hydrography basically. So temperature is shown on the color bar. So the color map is temperature. And then the blue contours, and I hope you can see these, show salinity. So we see a pretty classic doming up of the pycnocline in C1. So this is what you'd expect to see in a cyclonic eddy, this doming up in the center. Pretty much the same thing, but less pronounced in C2. And what I want to sort of draw your attention to is the skew towards the west. So we're not seeing a symmetrical sort of doming up as we do in C1. C2 is sort of a bit more skewed. And you can really see the warmer water in the surface of C2 compared to C1 over here. And then when we look at the anticyclonic eddy, we expect to see a sort of doming down of the pycnocline. We don't see that so much in the salinity contours, but if you look at temperature, you do see this doming down showing up in temperature. So this is not a super well-developed anticyclone, but it is showing some of the features that we would expect to see. Okay, so now I'm showing you fluorescence. So fluorescence, and then the white lines are density contours. So the previous plots I was showing you temperature and salinity. Now the color maps give fluorescence and the white lines give the density contours. Again, in C1, you can see this sort of classic doming up of those density contours. But then this feature I find kind of intriguing, and I'll come back to it, but we have a really high chlorophyll feature that's offset to the offshore side of C1, but we do generally see higher fluorescence sort of within the mixed layer of C1, not so much near the surface. Then as we move offshore into C2, again, we've got this sort of classic doming up of the pycnocline, but again, skewed off to the offshore side to the west. Chlorophyll again, or fluorescence, is enhanced in the mixed layer and a little bit more so at the surface than in C1, but much lower values that we saw in C1. So we're still seeing higher chlorophyll, but this is pretty spectacular what we're seeing in C1. And then in AC, again, it's sort of a pretty flat structure, not a huge amount going on. There's some doming down of the density contours that we can see here. And we have high chlorophyll in the mixed layer. But just for reference, these are on different scales. So this is going up to 3.5 for these two, but only up to 2.5 in the anticyclone. So we do see a little bit less biomass, less chlorophyll in the anticyclone compared to these two cyclones. And to me, AC and C2 look very similar. They look a lot like each other. C1, this younger cyclone, is sort of what looks much different to the other two, which maybe we would expect, because C2 has been hanging around for a long time. It's kind of getting old and tired and less less interesting than C1 maybe. Okay, and now I'm going to show you guys nitrate. So same thing, C1, C2, the white lines give the density contours. And then nitrate, this time, they're both on the same scale here. And we see really clearly how much the nucleocline has been domed up in that first young C1 cyclonic eddy. Really, really up into the top sort of 20 or well, 40 meters at least. And clearly we see that this interesting density feature is also reflected in the nitrate. So we have a sort of elevated nitrate feature along the inside edge of this younger eddy. And then moving on to C2, we clearly see that subduction of the nucleocline. So you can sort of see here, you're looking at, you know, nucleocline around 50 meters, it's dropped down to about 75 in that second cyclonic eddy. So there's quite a big difference in the vertical structure. And again, we see this sort of skew, this westward skew with more of a slope here and more of a gradient in nitrate on one side than the other. And then AC, again, it's actually not, I feel bad saying this, but the anticyclone is just not very exciting compared to the cyclones. We have a really flat structure. And the nucleocline in the anticyclone is actually much depressed compared to these two cyclones. So here you're looking at sort of, you know, 100 meters versus 75 versus 50 as an eyeball for the nucleocline depth. So again, you know, we're seeing quite different vertical structure in these features, but, you know, we're really looking at quite a small area of what's thought of as the transition zone as one regime. But if you look a little bit closer, you sort of see a lot of variability within that zone. Okay, so a quick recap. And one of the ways that I'm thinking about this, so I sort of have arranged these in a sort of an order, going from freshly upwelled coastal waters roughly, and then moving offshore into more oligotrophic waters over on this end. So I would say this C1, this coastal cyclone is, you know, it's derived from coastal upwelled waters that have been pinched off and form this cyclonic eddy. So this should look pretty close to what we would have seen in that upwelling plume. And then as it ages and moves offshore, we get into the C2, where we can see that the nucleocline has been subducted, much lower chlorophyll, so less productive. And then gradually it's going to start to look more like these anticyclones that are formed from the more oligotrophic California current waters, where the nucleocline is even further depressed, it's even deeper in the water column, and biomass is even lower. So, you know, my way of thinking about this is that we have this sort of time for space way of thinking about it. You know, we looked at all of we sampled all of these features at roughly the same time, but because we can track how they have formed and how old they are and roughly where they came from, it gives us a sense of how the evolution of this system might look in time. Okay, so that's sort of pretty much what I have looking at the biogeochemical properties. So we've got this sense of, you know, we've tracked these freshly upwelled waters that I've shown you here from the upwelling plume through these cyclonic eddies out into the anticyclone. And this, you know, this to me is a mapping of how those upwelled waters get processed through the transition zone as they are gradually moved offshore across that region. And we have some sense that, you know, the phytoplankton community is responding in terms of nutrient stress, responding in terms of community shifts, and how well and how efficiently it's able to reproduce, essentially. And again, we see this differential drawdown of the nutrients, which I think, you know, I still need to think about that some more, but that's feeding back into these community shifts that we're going to see. All right, so let's get into the phytoplankton community composition and how that's varying across these features. And so I'm just going to say again, this is work that Drew Chappell has spearheaded more so than me, and her master student, Susie Abdallah, who graduated this year and is now a Knaus Fellow, did a lot of the work on this. So this is coming back to C1 and C2. So remember I told you we did these CTD surveys, but then we also turned around and then did an underway survey where we collected water samples from the surface as we came back across this feature. And we actually completed this entire transect, and I think in roughly 24 hours. So this was another fun oceanographic sampling mission, quite a long day. So Drew collected samples roughly every hour from the underway system. So that was about 3 meters depth. We were traveling at roughly 10 knots. So these samples are about 20 kilometers apart, and filtered whole seawater onto 3 micron filters. So 1 to 2 liters for each of the samples. I did none of this work. So again, if I say something silly, it's my fault and not Drew's. And if you have any specific questions about the sequencing or the extraction, I probably can't answer them. So I apologize in advance. But she extracted DNA, and then that was amplified with diatom specific primers. So the goal here was to really target the diatom community in this analysis. And then sequences were analyzed using MED, and then those nodes were clustered at 99% similarity, which I know maybe a tiny bit about, but essentially that was how we got down to the OTUs and then matched them up to get at taxa. Okay, so this is what we found. And there's a lot on this figure. So I'm showing here the relative abundance as bar charts for each of the samples. And this is the color coding for the different species. So this gives you the relative abundance. And here is the dendrogram just showing how all of those samples clustered with each other. So this is based on the Bray-Curtis similarity matrix. And I think there are a couple of things to sort of note just straight off the bat. There is a cluster of samples here that are very much dominated by one species, which turns out to be Rhizoselenia. We have another cluster that seem to be much more diverse, but have a sort of, I'm not sure if you could say that Fragilariopsis dominates in every case, but it's an important component of the community in these samples. And then we have a sort of, you know, the weird mishmash samples that are doing some interesting things and are sort of a combination of these other two larger clusters, but it's not entirely clear how. But we are seeing some distinct clusters, and sort of just quickly mapping onto it here, there's this cluster that relates back to C2, and this one largely relates back to C1, but I'm going to talk about that in a bit more detail. So here's the map again. So this is the C2, the older eddy, C1, the younger eddy. These are all of the stations. And I've color-coded these two clusters from the dendrogram and then just put them onto the map. So we have a very clear separation of the community that we see here in this sort of pink C2 cluster compared to the community from this green cluster that maps back onto C1. And I gotta say, I thought this was really cool, actually, just seeing how distinctly these separated out in the dendrogram, that we have these, you know, geographically quite close together features, but in terms of the phytoplankton community, they're very different. And it's really demonstrated by this analysis. So then sort of getting into the details, now sort of digging into this cluster over here that I've coded in orange, this is a little bit less coherent. And now we're looking at a cluster that sort of relates more closely to the edges of these features. So geographically distant from each other, sort of across these different features, but in turn, well, ecologically more related. So I'm thinking that here we might be seeing some signature of advection around these features. So we might sort of just be seeing these communities that are being spread around the edges of the eddies, being quite different from the ones in the cyclonic eddies. And then again, we have this one sort of last quite different cluster. And again, you know, that's sitting in between along the edges of these two cyclonic eddies. So I think this work, I love it. I think it demonstrates really clearly that the diatom community at the surface, at least, is quite strongly structured by the flow field of these mesoscale features. Okay. So just a couple more things I wanted to draw your attention to. So that big shift we see between C1 and C2 really seems to be related to a shift towards rhizoselenia being completely dominant within C2, that older cyclonic eddy. So this plot shows you the relative abundance of rhizoselenia. This is C1, the younger eddy, and C2, and you just see this complete, you know, huge uptick to about 80% relative abundance within the second cyclonic eddy. And if we look at the diatom diversity across these features, so Shannon index is shown in the top panel, the evenness in the bottom panel, again, C1 versus C2. Not surprisingly, where rhizoselenia becomes so dominant in terms of the relative abundance, the Shannon diversity index just drops. So we're shifting from a relatively diverse feature in C1 and even in between the two eddies, but C2 as a whole is a much less diverse feature in terms of the diatom community. And that's reflected mostly through shifts in the evenness. So this suggests to me that actually the number of different OTUs that we've seen across the two features doesn't change all that much, but the way they're organized in terms of relative abundance is what's driving this big drop in the diversity. So it becomes much less even in that second older eddy. So Susie then,
Sophie,
you know, does some work.
Sophie,
Yeah.
This is Tatiana. Just a quick question
Mm-hmm.
before you move on. Did you just say that that richness was fairly similar across the transect?
It was, and actually I remember seeing a figure showing the richness that Susie made, that I asked her to make, but I could not find it before putting this talk together. But my recollection is that richness does not have, it doesn't vary a ton, but more so than how much it varies, there are no sort of, there are no distinct patterns in the richness that map back into the features, if that makes sense.
Thank you.
So Susie was interested in sort of trying to work out why rhizoselenia, like sort of why would that particular species really take off in this one particular feature? And so her thesis in the end was that the reason rhizoselenia could do so well in this older cyclonic eddy, which has a deeper nutricline that was quite oligotrophic in the surface waters, was that it has different strategies to access nitrogen that's not sort of readily available in the photic zone. And you know, we don't have any direct evidence to prove either of these hypotheses, but rhizoselenia can form vertically migrating mats. So they can actually drop down in the water column to get closer to the nutricline in order to access those deeper nitrogen pools. And we did actually observe when we moved across that second cyclonic eddy, C2, that the wind speed was quite low, and it was low enough that rhizoselenia mats could form. So environmentally, the sort of conditions were roughly right that they could have been forming vertically migrating mats. Another hypothesis, or another sort of potential explanation, was that rhizoselenia can host nitrogen fixing symbionts and form DDAs. And we were not, you know, we haven't got the data to test that hypothesis directly, but I think that's something that Drew certainly has been thinking about and will probably try and revisit at some point. So we have a couple of, hypotheses or potential explanations for why rhizoselenia may have really just picked up in that second, older, more oligotrophic cyclonic feature that we saw moving across the transition zone. Okay, so one quick last thing. So I talked about this sort of fine scale eddy edge effects. So this is what I'm interested in trying to work out, and I don't have a good explanation yet, but I'm really curious about these features here that we're seeing, very clearly in the density contours, and here very clearly in fluorescence on the westward sides of these two cyclonic eddies. And I'm not sure if this is a kind of advective pattern where we're really sort of just seeing, in this case, maybe more coastal waters have been wrapped around that eddy, or if there's some localized upwelling that might be, localized process that might be driving upwelling, maybe to do with the wind. So we see it really clearly in fluorescence here, and we also saw it pretty clearly in nitrate, particularly along this younger C1 cyclonic eddy. So that's, I have some ideas, but not developed enough to share with you guys yet. But I thought those, to me anyway, those features were particularly interesting. And it was a shame that we couldn't do the sort of traditional bow tie surveys across these features, because then we might have had a much better idea about this advection versus local upwelling question. Okay, that's it. That's all I have to show you guys. So just a quick wrap up slide. The way that I have been thinking about this, and my co-conspirators have been thinking about these data and this project, is that really what we've done is we've observed this succession in terms of the ecology of the phytoplankton community and the biogeochemical properties moving from this freshly upwelled water through these cyclonic eddies out into the more oligotrophic anticyclone. So we saw shifts in the nutricline, so it got deeper as we moved into the older features. We saw differences in the level of dissolved iron. We saw differential drawdown of silicate and nitrate near the coast that then sort of stabilized across the older cyclonic features. We saw a much more diverse, in terms of evenness, diatom community near the coast that became less diverse as we moved offshore. And we saw this really strong dominance of rhizoselenia in that older cyclonic eddy that we think might be related to the lower nutrient levels there. And then just sort of stamped on top of that larger scale, I guess, larger scale succession, we saw also some fine scale variability around these features. So at the surface in the diatom community structure, as well as some of these vertically resolved differences in the depth of the nutricline, and in where we saw those big upticks in the fluorescence, especially in that C1 eddy. So that's all I have. Thank you so much for giving me the opportunity to talk to you all about this. And also I want to again acknowledge all of my co-authors and sort of co-conspirators in putting this project together. And I would love questions or comments about this.
Thanks very much, Sophie. If people have questions, just go ahead and unmute yourself or type them in the chat if that feels more comfortable.
I might just. Shall I stop sharing my screen? Or at least, there we go.
Sophie, how do you figure out the age of the eddies?
So you can track the sea surface height anomalies, but there's also a really handy product that's been put together by Aviso, and I think it's what's it called? It's called something very unoriginal, like the mesoscale eddy product, where they actually identify and track different eddies essentially. I and they will track them through time. So they'll, for each one that's been sort of tagged, it'll give you the dates and location of formation. And it gives you other statistics like the size, the speed of rotation, and things like that. So very handy.
Yeah.
Hey, Sophie, it's Melissa. I have a question. So you showed this neat sort of feature in the, like this asymmetrical feature in the picnic line associated with, right there.
Yeah.
And I'm wondering, did you see that feature in the Aviso data? Like, was it associated with anything coherent in the sort of the spatial maps?
I don't remember seeing anything in the Aviso data, but actually I should take another look, because I imagine it might be sort of maybe all of the SSH contours might be closer together. So I'll take another look. That's a really good idea that I hadn't thought of.
Thanks.
Sophie, thanks for an interesting talk. I have a quick question about the species. So do you have any like water samples, anything fixed, or any like, I don't know, flow cytobot imagery that could help you test your hypothesis that Rhizoselenia or Fragilaria were dominating in the communities?
I would have to check. So I don't know of any fixed samples, because if we have any, Drew would have them. We did have someone on the cruise running FlowCam, but I haven't seen any of the data from the FlowCam, but presumably it is out there, so I could get in with, that was Tanya Peterson. So I might just double check with her.
And we,
Oh, sorry. Okay.
to put together a couple slides from the beginning and the end, you talked at the beginning about the nutrient patterns across the two eddies. And then in the end, you talked about the idea that perhaps, if Rhizoselenia is really dominating, and if it had cyanobacteria, it may be fixing nitrogen. And is there any way you can look at, let's say, N to P ratios or N to silica ratios to get a sense of whether nitrate, which is what you're measuring, or nitrite, whether inorganic nitrogen is being underutilized compared to N2? Like, is there a way to kind of get at, by looking at the ratios, whether it looks like there's too much biomass for the amount of nitrate?
Yeah, we have that data. I have nitric silica. I also have phosphate data. So yes, could certainly look at the ratios. I know that Susie did look at them, but I would have to refresh my memory a little bit about what the ratios were. I don't remember off the top of my head what we were looking at, but no, that's certainly a good idea. And it's sort of definitely something that we've done, but I just don't remember the details.
Thank you.
Hi, Sophie.
Hi.
So thanks for the talk. It was very interesting, particularly to see the biodiversity shift at such a small scale. And I was wondering, why did you focus only on the diatom community? And do you have an idea about the importance of the diatom in terms of phytoplankton community structure? And second question, you didn't want to show for the moment some flow cytometry measurement, but according to some preliminary result, do you see, for example, any kind of shift between Prochlorococcus and Synechococcus from the side and the middle of the edges?
I'm going to start with question 2, because that's one that I know much more about. Yes, so from this flow cytometry data, so first of all, just the overview, essentially, we see a lot of pico-eukaryotes near the coast. Gradually, Synechococcus takes over. So in C1, in C1, the pico-eucs were relatively abundant, but so was Synechococcus and Prochlorococcus. They were not hanging out in there. And then as we got into C2, the older eddy, then Synechococcus kind of took over from the picoeuchs, and Pro was still not really all that abundant, because remember, we're still in pretty cold waters in that C2 eddy. As soon as we got out of C2 into the oligotrophic California current waters, Prochlorococcus was everywhere. So that's the sort of larger scale view. At the edges of the eddies, yes. So we did, particularly at the edge of the offshore edge of C1, there was a big uptick that was associated with that, that nitrate, sort of shallow nitrate feature, and that DCM feature sort of off to the side of that eddy. So yes, we did see an uptick there. And I'm going to say, if I remember correctly, we also saw in Synechococcus particularly a big uptick in cell counts around the edges of C2 on the inside. So yeah, there was some fine scale structure in the phytoplankton that we see in the flow cytometry data. All right, first question, sorry, rewind. So why looking at the diatoms? All right, so the stupid answer is that my colleague Drew Chappell is an expert in diatom ecology. So that's one reason for looking at them, because that was her part of the project. But also diatoms tend to be a really important component of the community in these upwelling regions, because they can take up these upwelled nutrients pretty quickly and grow fast and sort of produce blooms based on those nutrient inputs. So just generally speaking, in this upwelling system, diatoms are a really important component of biomass and production. So that was another reason for focusing on the diatoms rather than the rest of the community. We do have actually other DNA samples that were extracted and that we sequenced with more general primers, 16S and 18S primers, to look at the whole community. But I don't know, I probably said this at the beginning, but this was kind of an ad hoc study, unfunded. It was one of those things where you're like, oh, well, we're here and we have stuff to take samples and there's this cool opportunity to make measurements, so let's do it. So it's, you know, we have this great data set, but there's also trying to juggle processing and analyzing some of these samples with other projects that actually are funded. So there are files that need to be run through the bioinformatics, but it just hasn't been done yet.
Thanks.
Sophie, can you see the chat?
I can't, no, I can't. If I stop sharing my screen, I think I'll be able to see it.
There's a couple questions in there.
Okay, wait a minute, let me. Zoom always confounds me because things disappear and then reappear, and I'm trying to like not share my screen anymore, but I can't find the thing that I can use to unshare. Oh. Ah, there we go. Right, okay, chat. Oh, smaller eddy, cool, I hadn't thought of that. I imagine it's possible. So I have, another thing I need to look at is the ADCP data. So there was ADCP data collected during the cruise, which I do mean to line up with the rest of it, especially for really identifying the edges of the eddies. So if there was a smaller feature that had been spun off from the edge of C1, that should be pretty easy to spot from the ADCP data across. So yes, I hadn't thought of that, and that would be a good idea. Species, okay, I think I've answered the rest of these questions. So in terms of other species, we have the potential to get data on things other than diatoms, but it hasn't been processed yet. So it's something I hope to do in the future, but it's just a question of time and manpower to do it.
Great, thank you. Thanks for a good discussion. Okay, if there are no more questions, postdocs and grad students stay on the call and we'll have a discussion with Sophie and everyone else. We will see you in 2 weeks for our next Bio at Noon.
Thanks, everyone.
Thank you.
Sophie, I have diatom community composition, growth rates, production rates for that upwelling plume section. So I'll send you the paper, and if anything looks useful, I can send you data and